And now let's jump in with an introduction to our first speaker, Dr. Fabian Graham. Fabian is a postdoctoral research associate in Dr. Ivan Rusin's lab in the Department of Veterinary Integrative Biosciences at Texas A&M University. His research focuses on the application of organotypic cell culture models in the development and implementation of high-content screening assays for predictive chemical safety assessments. Prior to joining Texas A&M in 2014, Dr. Graham was an NIH/NIEHS Superfund Research Program trainee and earned his PhD in human toxicology from the University of Iowa. Among other distinctions, Dr. Graham is the recipient of the 2015 Society of Toxicology Colgate-Palmolive Postdoctoral Fellowship Award in vitro toxicology, and was previously awarded the Golden Spatula 2009 for the best biochemical thesis in higher professional education in the Netherlands by the Royal Netherlands Chemical Society. Now I'll hand off to Fabian to begin his talk.
Well, thank you for the nice introduction, and I hope everyone can hear me. Okay, so welcome everybody to my webinar presentation. Today I would like to talk about how we can apply multidimensional high-content screening assays for toxicity profiling and ultimately predictive chemical safety assessment. I would like to begin with a brief introduction about current challenges and opportunities in the field of toxicology. And it's certainly a key problem that we're facing is that we have too many chemicals and too little toxicity data available for comprehensive regulatory decision making. So you can see here in the figure on the top left the numbers of chemicals that are listed in a variety of EPA inventories, thousands actually. And on the figure on the right, there's the percentage of available toxicity data for these chemicals. So altogether, you get the impression that there's only a fraction of these chemicals thoroughly toxicologically characterized. And this resulting chemical data cap is at least partially attributable to the insufficiency of traditional toxicity testing methods. There's still a requirement for in vivo data for regulatory decision making. However, as we all are aware, working with animals can be expensive and time-consuming, and is also generally associated with relatively low throughput. Traditionally, in vitro models such as cancer cell lines have been rarely included in regulatory decision-making, primarily because of the limitations in terms of physiological relevance. So altogether, there is a demand for novel in vitro and in silico approaches that make use of physiologically more relevant organotypic cell culture types, both in 2D or 3D applications, and that make use of new screening approaches as well, such as cellular imaging and various omics techniques. Considering the large data sets that we'll be generating, we'll rely more and more on computational data integration and visualization to ultimately break down these data sets to, communicable units. And a potential key application for high-content screening data lies in predictive safety assessments through read-across. Read-across is actually a fairly simple principle where when you have bioassay data available for a number of chemicals, and then you have data gaps for chemicals that have not been characterized. You can look for similarities, for example, a structural feature, and then based on these structural similarities, you can make predictions on the bioactivities. Such a chemical structure-based read-across has been utilized many times, both under the REACH initiative and also under OECD guidelines. However, ideally, you have access to more toxicity data for more chemicals so that you can actually approach a data integration-based read-across. And that is actually, it's a more sophisticated way of thinking about this. You have these toxicity profiles that you establish. On the right side here of the slide, represented by these pie charts. And in these pie charts, each slice represents an individual assay of toxicity endpoint, and the area of the slice is proportional to the relative toxicity of the chemical. So then you can categorize chemicals based on similarities in toxicity profiles. And the idea is then that that data-limited substances can be matched or not matched, for that matter, to a respective category, and thereby enabling the prediction of bioactivity. Such a biological data integration-based read-across approach may also be the only feasible option for highly complex substances that fall under the umbrella term UVCB. UVCB means unknown or variable composition, complex reaction products, and biological materials. And typically, these UVCBs are not amenable to chemical and structural characterization. Many of these UVCBs are high production volume chemicals. One may think of petroleum substances, for example. And they also face this amplifying complexity issue with batch-to-batch variabilities, because even though they're registered under the same CAS number and contain generally similar components, again, batch-to-batch variability is certainly something that cannot be prevented easily. The data integration-based read-across approach, particularly for UVCBs, is still at the hypothetical stage at this point, so case studies are needed to actually validate the applicability of in vitro and in silico assays for ultimately regulatory submission. This is an overview slide of what we're doing in our lab to tackle this issue. We're using high content screening assays in concentration response design, and we're currently screening induced pluripotent stem cell-derived cell types. We're focusing on cardiomyocytes and hepatocytes at the moment, but we're also looking to expand this spectrum to other cell types. The initial part typically consists of an assay development and endpoint multiplexing stage, where we test essentially the ideal combinations that provide us with the most data for the cheapest amount of money we can actually get, of course, to decrease the overall cost. All of these assays are typically have quantitative outputs, so we use these quantitative outputs for dose-response profiling to determine statistical parameters such as the point of departure value as an indicator of toxicity. We're also looking to include a high-throughput transcriptomics platform using a targeted RNA sequencing technology, and in order to slowly approach the chemical complexity of UVCBs, we're testing ion mobility mass spectrometry. Ultimately, all of these quantitative outputs need to be integrated into toxicity profiles, and that's the goal. We're using ToxPy software, which is available free of charge from the US EPA website, to integrate data into these pie charts I mentioned before, and where each slice represents an individual assay or endpoint, and the area of the slice is proportional to the relative toxicity. And then we want to use these toxicity profiles for substance categorization. In order to achieve this, we have collaborated with a number of government and industry institutions, including the National Toxicology Program, Concawi, and the Dow Chemical Company, with the ultimate goal being to test rapid, comparably inexpensive, and comprehensive approaches to increase the confidence in category-based predictive safety assessments. Today's webinar is going to focus primarily on the high-content screening assay development and multiplexing, as well as the data integration and categorization in ToxPy. I'm going to give you an example of one of the case studies we did. And well, this slide is an overview of a study that actually just got accepted for publication in Assay and Drug Development Technologies, where we tested essentially a variety of different endpoints for high content screening in induced pluripotent stem cell-derived cardiomyocytes and hepatocytes. For cardiomyocytes, we had two sets of experiments. The first one was a live cell screening, where we used the Flux instrument to measure the intracellular calcium flux of beating cardiomyocytes, which is an indirect measure of cardiomyocyte contractility. And then subsequently, we determined the intracellular cyclic AMP formation in these cells, which was used as an indirect measure of GPCR activity. In addition to this, we used fixed cell imaging using the ImageXpress XLS micro instrument with cardiomyocytes to test the probe combination for nuclei, mitochondria, and also for reactive oxygen species. Should probably mention that we're currently in the process of acquiring the new confocal version of the ImageXpress micro, so we're actually looking forward to the enhanced imaging capabilities for follow-up studies. For hepatocytes, we used the same probe combination as for cardiomyocytes, but in addition, we also included an actin stain for cytoskeletal integrity, and we tested a neutral lipid stain for intracellular lipid accumulation in hepatocytes, sort of an in vitro steatosis assay. Let's go a little more detail about the calcium flux. As I mentioned, these cardiomyocytes form a beating monolayer in culture, and the intracellular calcium flux is an indirect measure of their contractility. So in untreated cells, as shown here on the top left, you can see this regular, nice beat pattern of this calcium flux. Our vehicle control did not show any effects, whereas cytotoxic concentrations of chemicals are typically resulted in no observable peaks. Kind of makes sense. But most importantly, the calcium flux assay is capable of reproducing drug-associated phenotypes in vitro. So you can see the QT elongation, for example, as well as increases or decreases in the beat frequency. And more importantly, we do not only have the phenotypic qualitative measures, but we also have quantitative data outputs that give us data on the peak count, frequency, the average amplitude or peak spacing, and then many more actually that we can use subsequently for dose response profiling. In this particular study, we measured calcium flux for up to 90 minutes, and then we lysed cells in order to determine the intracellular cyclic AMP concentration using the catchpoint assay kit. This assay kit it's a very simple principle. It's in fact a competitive ELISA, and how it works is that with increasing intracellular cyclic AMP concentrations, you quench the background signals. I can see that in the standard curve on the left here, and in the figure on the right, you can see essentially that untreated cells have a baseline fluorescence level, and then when you stimulate cells with a positive control, for scolin in this case, you decrease and suppress the fluorescent signal actually down to background levels. We started this experiment out with a time course study looking just at addition of media or DMSO to cells, and you can see here also on the top left in panel A that after 10 minutes, cells appeared to be very irritated. There was high variability and actually a decrease in beat rate observable even with just addition of media. But already after 30 minutes, they appear to have mostly recovered to, still with a higher variability, but after 60 and 90 minutes, the readouts were really solid. When we looked at drug-associated phenotypes, I should probably mention that this figure here on the top is really one representative parameter only. It shows the cardiomyocyte beat frequency. And our positive control for increases in beat frequency was isoproterenol, and that was also the drug that gave us the only drug that gave us a nice concentration-dependent increase in the beat rate. So both after 30 minutes and 90 minutes. When we looked at cyclic AMP formation, isoproterenol was also the drug that induced by far the most potent increase in intracellular cyclic AMP levels. Again, this cyclic AMP or GPCR activity assay is fairly non-specific, so that also explains why we saw some fluctuations even with drugs that were not associated with increases in the beat rate. However, in order to test this in a, I guess, quote-unquote real-world setting, we screened a number of gas oil extracts using this assay combination, and what we found was actually very nice concordance between the increase in beat rate and increase in intracellular cyclic AMP levels. So there is an indication that potentially GPCR activity is involved in these cardiophysiologic changes. But obviously, in order to, there need to be follow-up studies to dig deeper into the mechanism. When we look at cell imaging and the three probes that we were using, as mentioned before, the reactive oxygen species, nuclei, and mitochondrial probes, we saw a very interesting time course development, because after 30 minutes we were capable of detecting reactive oxygen species following treatment with menadione, doxorubicin, and tetractylammonium bromide. However, these effects were gone after 24 hours. By contrast, cytotoxic effects and mitochondrial effects were observable after 30 minutes, but they were a lot more pronounced after 24 hours, so it was a distinctive pattern. For hepatocytes, we observed the same pattern for this particular experiment. We were capable of detecting reactive oxygen species following treatment with menadione after 30 minutes, but also these effects were gone after 24 hours. And also here, cytotoxicity was apparent after 30 minutes, but was more pronounced after 24 hours. And when you look at, actually, when you stretch this out to the 48-hour time point, it's shown here on the right side of the slide, the cytoskeletal integrity as a measure of cytotoxicity shows that after 48 hours, the concentration response curves are even further pronounced. So time course development. When we look at the lipid accumulation, we used amiodarone is a positive control for that particular assay. Amiodarone is a known steatotic agent and has been used as a reference control in steatosis assays. And it was also the compound that gave us by far the best concentration-dependent increase in intracellular lipid accumulation. So again, overall, similar trends observable in both hepatocytes and cardiomyocytes. However, I would now like to introduce you in how we can actually apply these high-content screening assays for actual case studies and industry applications. This particular collaboration with Konkawi is a consortium representing major oil companies in Europe, and they are coordinating environmental research for them. Konkawi was kind enough to provide us extracts of petroleum substances, and petroleum substances are a particularly interesting case study for us for a number of reasons. First of all, petroleum substances, and depending on the carbon chain length, there can be up to billions and actually trillions of theoretical isomers in these substances. They are prototypical high production volume chemicals, so they do matter. We're exposed to them in many parts of our everyday life, so we want them to be safe. Technically, interesting is that they are chemically highly diverse. So crude oil is initially processed by fractional distillation, which results in all of these obtained fractions being very, very differing and distinguishable based on boiling point and volatility, but also on molecular weight and carbon chain length for all these different product classes. And then obviously, what I mentioned earlier in this presentation, the batch-to-batch variability is certainly an area of interest that can potentially be addressed using high-content screening assays. So the working hypothesis for this case study was that petroleum substances of a distinctive product group can be categorized for read-across applications based on physical-chemical properties, but that we can also increase the confidence on this grouping using actual bioactivity fingerprints and outputs. This is a very simplified overview of the oil refining process. And again, the first part is based on fractional distillation, but then there are a number of conversion and upgrading processes. I don't really want to go into too much detail here, but I guess the key message is that we received 26 extracts of petroleum substances from a variety of different product groups that really span almost the entire spectrum of this oil refining. So we did have a highly diverse group of samples to work with. Experimentally, we used calcium flux screenings in live cardiomyocytes, as well as a combination of cytotoxicity measurements via imaging in both cardiomyocytes and hepatocytes. All the quantitative outputs were normalized to control wells before we conducted dose-response profiling in R, and then we used the derived point of departure values as inputs for toxicity profiling and Toxicology. I will talk about this second part in more detail in just a few slides, so bear with me. On this slide here, you can see essentially an overview of category-specific effects of petroleum substances on cardiomyocytes using the beat frequency as a representative example parameter. So again, we have these types of curves for a variety of assays and parameters. But the take-home message here is really that within a certain product group, the concentration response curves really show a high degree of similarity, even though the potency may differ somewhat. But that is not necessarily the case between groups, so there are pronounced differences between those groups. And you can also see, again, like what we showed before, this biphasic character for some of these curves, where we have initial increases in the beat rate followed by sharp decreases, which are typically due to cytotoxic concentration. For hepatocytes, the overall conclusions from looking at these concentration response profiles were pretty much similar. So there was a high degree of resemblance within a product group, and there were pronounced differences between product groups, even though some of them also resembled each other. We'll talk more about that in just a bit. The data integration using Toxicology was again based initially on dose response profiling in R. We fitted data points to a nonlinear function and then determined point of departure values, which are in this case defined as the intersection of the fitted curve with one standard deviation around the means of the control. So it's basically a measure of the onset of toxicity. And the way this works now is we scale point of departure values from 0 to 1, and the lowest observable point of departure value, which indicates the highest relative toxicity, was assigned the value 1, and the highest point of departure value, which is associated with the lowest observed toxicity, was given the value 0. And then we used these scales, Toxicology values, I guess, for as data inputs for toxicity profiling. And then we used these pie charts essentially to attempt categorization of substances. I should also mention that Toxicology provides a final Toxicology score that integrates all of the areas of the different slices. And when we rank chemicals according to their Toxicology score, we came up with this figure, which was sort of an eye-opener for us because it really showed very clearly how biological data can actually support the physical-chemical-based categorization grouping that was hypothesized. So you can see that the vacuum and hydrotreated gases, for example, were found to be the most bioactive ones in this particular study. And the straight-run gas oils were close second, and these are related groups of petroleum substances. By contrast, the residual aromatic extracts and bitumens, for example, are found at the low spectrum of the bioactivities, and they were also clustered together. A notable exception were these Konkawi 19 and 20, as you can see on the top, that form sort of a separate little subgroup in this analysis, with more pronounced hepatotoxicity. This was also highlighted by correlation heat map analysis, where also we found very good clustering of these product groups, with Konkawi 19 and 20 being the notable exception. So in summary, we've developed a rapid and comparably inexpensive high-content screening platform that can be utilized for multidimensional toxicity profiling using induced pluripotent stem cell-derived cell types. And we have used this approach to biologically categorize petroleum substances. We also believe that Toxify-enabled data integration really provides an effective means of communicating complex data sets in fairly easily understandable units to potentially risk assessors and other stakeholders. Of course, we hope that this is just the beginning, and we're looking to extend this approach to further increase the confidence in the read across of UVCBs. And of course, we're looking at the high throughput transcriptomics and the chemical characterization using eye mobility mass spectrometry. So we're also looking at including a population variability type of experimental design, where we're planning to use cardiomyocytes derived from more than 30 different individual donors from at least 3 ethnically distinct populations. And then additional tissues, we would like to expand our tissue and cell type portfolio to also increase the confidence there and include additional toxicity data points. With that being said, I would like to thank my colleagues from Texas A&M University, as well as our collaborators from Molecular Devices and Konkawi for all the support we got, as well as Fred Wright and his group from NC State University for statistical support. I would also like to acknowledge funding by the Society of Toxicology, as well as the US EPA, and of course I would like to thank everyone in attendance for listening. Thank you very much.